Welcome to the fascinating world of microorganisms, where hidden life reveals its visual impact. In this journey, we'll take a closer look at the unseen yet powerful world of microbes. Microorganisms, defined as life forms too small to be seen with the naked eye, were first observed by Anton van Leeuwenhoek. These tiny entities are found everywhere, existing in various forms and sizes, and play diverse roles in our world. While some are beneficial, others can be harmful pathogens. Microorganisms can be classified based on their cellular structure and nuclear material. They range from non-cellular viruses to complex multicellular organisms like fungi and algae, each with unique characteristics. Microorganisms are divided into prokaryotes and eukaryotes based on their nuclear material. Prokaryotes, like bacteria, lack a nucleus, while eukaryotes, such as fungi and algae, have a defined nucleus and organelles. This distinction is crucial in understanding their biological functions. The microbial world is diverse, encompassing bacteria, viruses, fungi, protozoa, algae, and archaea. Each type plays a unique role in the ecosystem and impacts our lives in various ways. Now, let's move on to explore the different types of microorganisms. We'll begin with the first and perhaps the most well-known group, bacteria. Bacteria are unicellular prokaryotic organisms capable of independent existence. They reproduce rapidly and have a complex structure that includes an outer layer, internal structures, and appendages essential for their survival. The outer layers of bacterial cells, including the capsule, cell wall, and membrane, provide protection and regulate the flow of substances, and inside these layers the protoplast is present, which contains ribosomes, genetic material etc. These layers are vital for the bacteria's interaction with their environment. Gram-positive bacteria have a thick peptidoglycan wall for support and ion transport. It lacks an outer membrane, have little to no periplasmic space, and appear purple after gram staining. This structure provides structural support and plays a role in antibiotic sensitivity. In contrast, gram-negative bacteria have a thinner peptidoglycan layer, an outer membrane with lipopolysaccharides, a clear periplasmic space. They appear pink after gram staining and are generally more resistant to antibiotics. The bacterial cell membrane is a selectively permeable barrier composed of phospholipids and proteins. It controls the movement of substances in and out of the cell, maintaining the internal environment. In addition to the outer layers, bacterial cells also contain internal structures in protoplast and other external appendages like pili and fimbriae. Bacteria can be classified by shape, arrangement, structure, and cell wall composition. This classification helps in identifying and understanding their diverse roles and characteristics. Gram staining is a method used to classify bacteria based on their cell wall properties into two groups, gram-positive and gram-negative. The process involves applying a primary stain, a mordant, a decolorizer, and a counter stain. Gram-positive bacteria retain the crystal violet stain and appear purple. Gram-negative bacteria lose the crystal violet during the alcohol wash and take up the counter stain, appearing pink or red. Acid-fast staining is used to identify bacteria with waxy cell walls, such as Mycobacterium species. Acid-fast bacteria keeps the red stain after acid-alcohol treatment, while non-acid-fast bacteria lose it and appear blue or green color. Bacteria can be both friends and foes. They are used in food production and medicine but can also cause infections and spoilage. Understanding their dual nature is key to harnessing their benefits while mitigating risks. Bacterial infections begin with entry into the host, followed by adherence, colonization, and rapid multiplication. This process can lead to tissue damage and disease, highlighting the importance of understanding bacterial pathogenesis. The cycle of bacterial infection involves invasion, multiplication, and spread within the host. Understanding this cycle is crucial for developing strategies to prevent and treat infections effectively. Following bacteria, the second major group of microorganisms we'll discuss is viruses. Viruses are acellular entities smaller than bacteria, lacking organelles and relying on host cells for replication and has no flagella, making it non-motile. Their unique structure and parasitic nature make them fascinating yet challenging to study. The structure of viruses includes a capsid, genetic material, and sometimes an envelope. These components protect the virus and aid in host cell attachment, playing a critical role in viral infection. Viruses are classified by host type, shape, envelope presence, and genetic material. This classification helps in understanding their diversity and developing targeted treatments. Bacteriophages, also known as phages, are fascinating viruses that specifically target and destroy bacteria. They possess a complex structure, with each part playing a crucial role in their function. The head, or capsid, contains the genetic material, usually DNA, while the tail helps attach to the bacterial cell. Tail fibers recognize and attach to specific bacteria, and the sheath contracts to inject DNA into the host. This intricate design makes bacteriophages powerful tools in combating bacterial infections. Viruses are a double-edged sword, serving as both friend and foe. On the beneficial side, they are used in vaccines, gene therapy, and genetic engineering, with bacteriophages playing a role in killing harmful bacteria. However, viruses can also be harmful, causing diseases like flu, HIV, and COVID-19. They are highly contagious, can lead to chronic illnesses or cancer, and antibiotics are ineffective against them. Understanding their dual nature is key to harnessing their potential while mitigating risks. The pathogenesis of viral infections is a complex process. Viruses enter the body through infected persons, droplets, or fluids, targeting the respiratory tract, mucous membranes, and blood. They bind to specific receptors on host cell membranes, hijacking the host's machinery to replicate viral particles. This leads to cell death, immune evasion, and systemic symptoms. Moving on to the next class of microorganism, the fungi. The fungi are unique eukaryotic organisms, distinct from plants and animals, often recognized as mushrooms. They are non-motile and non-photosynthetic, with most being multicellular, though yeasts are unicellular. Fungi are closely related to animals, as genetic studies reveal, and are absorptive heterotrophs, secreting enzymes to absorb nutrients. Their structure includes chitin cell walls and thallus bodies, forming mycelium. Fungi thrive in warm, moist environments, playing a vital role in decomposing dead matter. Fungi possess key structures that enable their growth and nutrient absorption. Hyphae are thread-like filaments that absorb nutrients, with septate hyphae containing cross walls and aseptate hyphae lacking them. Mycelium forms the dense network that feeds the fungus, increasing surface area for nutrient absorption. Fungal cell walls are made of chitin, providing strength, while fungal cells may contain typical eukaryotic organelles like mitochondria. These structures are essential for the survival and function of fungi. Fungi are classified into various groups based on their hyphae and spore types. Zygomycota, Ascomycota, Basidiomycota, and Deuteromycota are the main categories, each with distinct characteristics. For example, Zygomycota have aseptate hyphae and zygospores, while Ascomycota have septate hyphae and ascospores. Understanding these classifications helps in identifying and studying different fungi, revealing their diverse roles in ecosystems and their impact on human activities. Fungi, like viruses, have both beneficial and harmful effects. They are used in baking, brewing, and cheese making, and are sources of antibiotics like penicillin. Fungi decompose organic matter, aiding in nutrient cycling, and play roles in biotechnology and enzyme production. However, they can also cause skin infections, spoil food, trigger allergies, and damage crops. Recognizing their dual nature allows us to utilize their benefits while managing their adverse impacts. Fungal infections occur when fungi enter the body through skin or inhalation. They attach to host cells, releasing enzymes that damage tissues, and evade immune defenses using biofilms or capsules. This leads to disease through direct invasion, allergic reactions, or toxin production. Understanding the pathogenesis of fungal infections is crucial for developing effective treatments and prevention strategies, as fungi can cause significant health issues. Proceeding to the next group of microorganisms, we have the protozoa. Protozoan cells have specialized structures that define their functions. The plasma membrane protects and defines the cell boundary, while the cytoplasm contains organelles and stores nutrients. The nucleus holds genetic material. Contractile vacuoles regulate water balance, and pseudopodia aid in movement and feeding, while flagella and cilia enable locomotion. Protozoa are classified into groups based on their movement and feeding structures. Sarcodina use pseudopodia, Mastigophora have flagella, Ciliophora use cilia, and Sporozoa are non-motile parasites. Each group has unique characteristics and examples, such as Entamoeba histolytica in Sarcodina and Giardia in Mastigophora. Understanding these classifications helps in studying protozoan diversity and their ecological roles. Protozoa helps in recycling of nutrients, feeding aquatic life, and supporting soil health. However, they can also cause diseases like malaria and amoebiasis and infect humans, animals, and plants. So far we have covered bacteria, virus, fungi, protozoa. Now we are moving to algae. Algae are simple, plant-like organisms, either unicellular or multicellular, and are photosynthetic, containing chlorophyll. Found in aquatic environments, they are mostly non-infectious but can produce toxins, such as those causing red tides. Algae can also trigger allergic reactions to spores and cause skin irritation from algal blooms. Despite these challenges, algae play crucial roles in ecosystems, contributing to oxygen production and serving as food sources. Algae are classified based on their pigments and examples. Green algae contain chlorophyll A and B, while red algae have chlorophyll A and phycobilins. Brown algae possess chlorophyll A and C, along with fucoxanthin, and golden algae have carotenoids and xanthophytes. Blue-green algae contain phycocyanin and chlorophyll A. These classifications help in understanding the diversity of algae and their ecological roles. Next, we move on to the archaea. They are ancient, single-celled prokaryotes, distinct from bacteria, thriving in extreme environments like heat, salt, acid, or methane-rich areas. They lack peptidoglycan in their cell walls and have no proven direct infections. Archaea are present in human microbiota, especially in the gut, mouth, and skin, playing roles in gut health and digestion. Their potential involvement in inflammatory diseases is still being researched, highlighting their mysterious nature. Archaea are classified into groups based on their habitats. Thermophiles thrive in hot springs and hydrothermal vents, while halophiles prefer salty lakes and salt mines. Methanogens are anaerobic, found in guts, and acidophiles inhabit acidic environments. These classifications reveal the adaptability of archaea to extreme conditions, showcasing their unique survival strategies. Archaea, while not directly causing infections, are present in human microbiota, especially in the gut, mouth, and skin. They may play roles in gut health and digestion, and their involvement in inflammatory diseases is still being explored. As research continues, understanding the balance between their beneficial and potential harmful effects is crucial for harnessing their capabilities. Microorganisms are responsible for a variety of infectious diseases. Bacteria cause illnesses like tuberculosis and cholera, while viruses lead to influenza, COVID-19, and measles. Fungi can result in athlete's foot and ringworm, and protozoa are behind diseases such as malaria and amoebiasis. Recognizing the diverse impacts of these microorganisms is essential for developing effective prevention and treatment strategies. Microbial infections follow a general mechanism, starting with a source or reservoir found in contaminated food, water, or animals. Transmission occurs through ingestion, insect bites, or contact with infected individuals. Microbes adhere to host tissues, invade cells, and multiply, leading to tissue damage and symptom development. Understanding these mechanisms is key to preventing and controlling infections. Microbial transmission occurs through various modes, including direct contact, indirect contact, airborne transmission, food and water contamination, and vector-borne routes. Direct contact involves person-to-person interactions, while indirect contact includes shared objects. Airborne transmission spreads pathogens through droplets, and contaminated food or water can lead to infections. Vector-borne transmission involves insects like mosquitoes. Recognizing these modes helps in implementing effective prevention measures. Preventing microbial infections is crucial for maintaining health. Regular hand hygiene, using soap or alcohol-based sanitizers, is a simple yet effective way to reduce the spread of germs. Vaccination keeps us protected against viral and bacterial diseases. Personal protective equipment, like masks and gloves, is essential in healthcare in crowded settings to minimize exposure risks. Safe food and water practices ensure proper cooking, clean drinking water, and safe food handling. Lastly, environmental sanitation involves disinfecting frequently touched surfaces to maintain cleanliness and prevent infection. Now on summarizing various microorganisms, we have that bacteria are prokaryotic, while viruses are acellular, and fungi and protozoa are eukaryotic. Their cell structures vary, with bacteria having a cell wall and cytoplasm, viruses having a protein coat, and fungi and protozoa having more complex structures. Sizes range from microscopic to visible. Reproduction methods differ, with bacteria using binary fission, viruses needing host cells, fungi using spores, and protozoa reproducing asexually. They cause various diseases, but also have beneficial uses, like gut flora in bacteria and decomposition in protozoa. Transmission methods include air, water, and vectors. Antiseptics and disinfectants play a vital role in destroying microorganisms on contact. Antiseptics are used on living surfaces, such as skin, to prevent infection, while disinfectants are used on non-living surfaces to eliminate harmful organisms. Inanimate surfaces like surgical instruments require sterilization to ensure they are free from pathogens. Germicides are common terms for both antiseptics and disinfectants, emphasizing their role in killing pathogenic and non-pathogenic microorganisms. Antiseptics are classified into various types based on their chemical composition and usage. Phenol derivatives like phenol and cresol, oxidizing agents such as hydrogen peroxide, and halogens like iodine and chlorine are commonly used. Biguanides, quaternary compounds, alcohols, aldehydes, acids, metallic salts, and dyes each have specific applications in preventing infections. Understanding these classifications helps in selecting the appropriate antiseptic for different situations. The microbial world is vast, encompassing bacteria, viruses, fungi, protozoa, algae, and archaea. These microorganisms are integral to ecosystems and biotechnology, providing benefits like decomposition and nutrient cycling. However, some microorganisms can cause diseases, highlighting the importance of understanding them for developing prevention and treatment strategies. Continued research is crucial to harness their potential for human health and environmental sustainability.